this is going to be like a cool, long presentation.
Yeah.
But everything is going to be awesome. 100 slides.
We'll see how it goes. Yeah, so they can hear you, otherwise I'm not here.
Okay, can you hear me? Can you hear me? No. Hello? Hello? Is it on?
I think they can hear you, right?
Can you hear me?
Well, I can hear you.
Yeah, because I'm shouting.
People in the back, I think. Yes, okay, so let's hear.
Hello? Hello? Hello?
Yeah, I think you're good. Yeah.
Okay. Okay, we're going to get started. Please take your seats. I'm very excited about this talk. Our seminar speaker today is Margarita Lopez-Uribe. Margarita is an associate professor and extension specialist at Penn State University. She got her bachelor's in Colombia, then went to Brazil to do a master's. After that, she came to the US to do her PhD at Cornell University. She did a postdoc at NC State and then joined the faculty at Penn State. Margarita, she looks at human-driven effects on wild bees. She looks at the effects of domestication, has looked at the effects of climate change, the effects of management practices, and how all those can affect wild bees. And so we have some things that we have in common. I'm interested in similar questions, but with tritrophic interactions. So without further ado, thank you, Margarita, for coming.
Thank you, Cesar. Thank you for the invitation. I must say I feel energized after the lab meeting with Cesar's group. We had a great discussion. I need to figure out how to put this on. Jeez. Wait a second. Okay, I think I figured it out. Well, thank you for coming today. Thank you, Cesar, for the invitation. I think this is what makes a difference between the virtual talks that we had for years during the pandemic and coming in person to interact with people, really get to know people. So, well, I'm going to talk about what we do in the lab with a focus on one part of the research program, but I will present a little bit of everything. And we really are kind of an ecology and evolution lab, and we work a lot in human modified environments. I wanted to start by giving a land acknowledgement. I work at Penn State University, which is a land-grant university located in the ancestral land of the Susquehanna people. And also introducing my lab. So this is currently my group. We have a huge diversity of people with different backgrounds and interests, but we're all working on different aspects of the ecology and evolution of bees. Cesar said, I have an extension appointment at Penn State, so some of the work that we do involves applied research. It's not only kind of fundamental research, and so I'm just going to go, give a brief overview of some of the things that we're working on, and then I'll dive into the main topic of today's talk. So we have a pretty sizable research and extension program on organic beekeeping. This is work that I have been working on with Robin Underwood for several years. So we've had funding from the organic program from USDA. And the goal of this project, of this program, is to support organic beekeeping in the United States. If you like honey and you support organic agriculture, you probably know that there is no organic honey that is produced in continental US. And this is basically what we're working on. There are some limitations and challenges about the standards for organic beekeeping. And basically we're trying to, pave the path so that we can support organic beekeeping in the US, continental US. We also have a growing line of research on collections and bee monitoring. So about 3 years ago, we started this project called the Pennsylvania Bee Monitoring Program. And I think this is one of those projects that is inspiring. I'm very passionate about what we're doing. It's a great collaboration between some members of our lab, David Bittinger, who is another professor in entomology, and extension educators at Penn State. So for the past 3 years, we have been working on developing a better understanding of the diversity and the distributions of bees. And we have been doing this in collaboration with master gardeners. So those are 20 people who have been part of this program for 3 years. And those, we train them to collect bees twice per month using netting and traps. And then they send us all of those specimens, and we're doing the identifications, and we're databasing all of this information. So this has allowed us to increase our kind of like knowledge, baseline knowledge of what we have in the state and where those bees are. And the hope is that we will be able to generate data to detect changes in things over time, both in terms of distribution and abundance, but also to improve, you know, like the knowledge of what's there. So, you know, like some numbers, we've had, the first 2 years of the project, we added like 600 new county records, 8 new state records, and all of these bees were collected by master gardeners. And of those 8 new state records, we have 3 species that are actually non-native from the US. So another goal or another thing that we're achieving with this program is kind of like this detection of arrival of things that we're not expecting to find, but you know, like that are showing up. We're also doing some work in, with environmental stressors and bee health. And so this is, I'm not going to dive into this, but this is a project in collaboration with Ruth Schilder, who is at Penn State, and Shalini Jha, who is at UT Austin. And basically we are working on interactions between stressors, but in the context of changes in temperature and humidity. Basically, you know, how all of these environmental stressors are interacting and really impacting or not bee health. Well, and finally, this is another line of research. It's very different, but we are working on ecoevolutionary dynamics of pollinators in cropping systems, and so this is what I'm mainly going to talk about today. I see some comments in the chat. Yeah, the camera is pointing towards the ceiling. I have no control over that. You know, like, I'm too short for this camera, so my apologies. Okay. Well, I wanted to start by giving you guys a little, you know, a short description of the framework of the context in which we're asking these questions. So we know that agriculture is a major driver of environmental change, and we know that the expansion of agriculture has been associated with biodiversity losses. This has been, you know, like demonstrated in bees, right? So we know that the agriculturalization of landscapes is associated with losses of a species. In this paper, Rab and collaborators actually demonstrated that for every 10% of agricultural intensification, you lose about 35 million years of, you know, evolutionary diversity in the communities around agricultural areas. So we know that there are losses associated with agriculture. But with everything in terms of the environmental change, we have these winners and losers in the context of these changes, right? A lot of the species are lost, but we see some species that persist very well, or that can even like thrive in environments that are modified by humans. And so kind of understanding what predicts these winners and losers can be, you know, like mainly driven by 2 mechanisms, or this is how I think about it. I would love to, you know, hear everyone else's thoughts. In this paper, for example, Rader et al. Try to identify life history traits that were associated with, you know, winners and losers. And so for the bee literature, we know that, for example, diet specialization is generally associated with the species that are lost when there is land use intensification. And so basically we can think of these as traits that make bees pre-adapted or not to these changes in the environment. But there is another mechanism, right? And so I think this is the question that I would like to pose today. Can pollinators actually adapt? to agricultural environments, right? Agriculture has been around us for many years. In the system that I work with, we started domesticating plants about 10,000 years ago. So a lot of time has, probably allowed, it is possible that it has allowed adaptive processes to take place in pollinators. Well, but then if we think about, okay, are bees adapting to agricultural environments? What are the potential drivers of selection, right? So I think of this in several ways. Again, this is how I think about it. I would love to hear, a lot of people here in this audience are thinking about domestication, so I would love to hear your thoughts. Well, in the first instance, we have changes in functional traits of plants, right? So we know that the structure of the plant community is an important driver of, how all the biodiversity is supported and associated in an ecosystem. And we know that crop domestication has changed the traits of the plants and they have been selected for traits of agricultural importance or agronomic importance, right? So here I have a diagram, we tend to select for sweeter fruit that are bigger. And guess what, right? Like the fruit are developing from the flowers. So there are reasons to think or predict that there are unintentional consequences of crop domestication in the functional traits of flowers. How is that impacting pollinators? We don't know. There are also consequences in terms of novel biotic interactions. So something that has happened since, many years ago is that once the plants are domesticated, they are introduced into different environments. And then the biotic interactions in these systems completely change, right? So how are, for example, for pollinators, right, like they are interacting with managed bees in these agricultural systems. How are these novel biotic interactions impacting wild pollinators? And last, there are kind of unique characteristics of agricultural environments. This is what we call kind of a niche construction framework. There are certain ways in which agricultural settings have common practices that make the abiotic conditions similar. So we can think about, for example, the microclimate, how open agricultural fields are, the exposure to xenobiotics. So yeah, again, this is likely another driver of selection in pollinators, and this is beyond pollinators, right? Like anything that is inhabiting agriculture. So well, about 3 years ago, I received an NSF career grant to work on these questions. So the broad questions that we have are around the evolutionary consequences that crop domestication and the expansion of agriculture has had on pollinators. So I'm going to talk a little bit about what we have done, but I first need to introduce you to the system that we have been using to study those questions. So we work with squash bees and cucurbita. These are the cute squash bees. And squash bees are they are a group of about 22 species. They are all narrow oligolectic. I know oligolectic is a word that not everyone may be familiar with, but what that means is that they feed on a very narrow number of plant sources. In this case, squash bees are actually monolectic. They only feed on the pollen of one plant genus, which is the genus Cucurbita. The bees are endemic from the New World. So this is a map pulled out of Discover Life, where you can see that all of these 22 species are found throughout North, Central, and South America, and they have the highest diversity in Mexico and in Central America. We've been working on understanding the evolutionary history of this group, so we built this phylogeny. This is not published yet, and this is allowing us to do some comparative work in the group of squash bees and relatives. And we know that these bees originated about 6.5 million years ago, which is about 5 million years after the origin of the host plant Cucurbita. But well, what makes this system really unique and interesting for the questions that I have for today is this relationship with the plants, Cucurbita. So this is a genus of plants. It's also from the New World. It originated in Mexico. And what is very interesting is that it is a plant genus that has been domesticated multiple times. So we have 6 independent origins of domestication in these plant genus. And so this allows us to do really nice comparative work to work on questions related to domestication, because we can actually see if there are expected repeated patterns of evolution that are associated with domestication. And they are agriculturally important, they are culturally important. So it's a kind of like a perfect system to ask the questions about the impacts of domestication and agricultural intensification on pollinators. There are other aspects that make this project very interesting and unique to ask these questions. One is that all of these plants are monoecious. That means that they, in the same plant, they have both female and male reproductive organs, but they are located in different flowers. So these plants must absolutely interact with biotic agents that are moving the pollen from the male part of the flower to the female part of the flower. And the flowers, of course, produce a lot of floral resources, right? So these are just some photos showing you the amount of nectar that sometimes you can find in Cucurbita flowers. So I don't know if you can see kind of like that pool of nectar at the base of the flower. And it produces a lot of pollen that is actually very large, spiky, and sticky. And so that actually makes the pollen not very palatable to some bee species, except squash bees. So when we look at the pollination systems, I want to kind of like wrap up this introductory part of the talk, saying that the plants, from the plant perspective, they actually are pollinated by various groups of bees, right? The plants are not specialized on any type of pollinator. Here we can see some stingless bees. This is a photo in the tropics. They're very common pollinators of Cucurbita in Central and South America. And that's a photo in Pennsylvania of bumblebees. So they are very popular flowers to many pollinators. And so when we think about what this means evolutionarily, it means that the specialization of the squash bees and Cucurbita is asymmetric, right? The plants are generalists, the bees are highly specialized on the plants. Well, for the rest of the talk, I am going to focus on this one species of a squash bee. The name is Cynoglossa pruinosa. So we're going to be talking about this one species, even though there are more out there that I would love to study. And this species is very interesting because it is the most widespread squash bee in North America. So this is data from Discovered Life again, and this is kind of an old map. I think there are probably more records now. But the point here is to show you that the species is very widespread in Mexico, throughout the United States, and it gets to like southern Canada on the East Coast. Now, what is really interesting is that there are no native cucurbids in the temperate zones of North America, right? So the most abundant Cucurbita wild species is really restricted to the deserts of the Southwest and kind of like the, like Midwest here, and then, like some more mesic habitats of Mexico. But we can see that there are lots of places in North America where they don't have wild plants that coexist with them. As I said, these bees are monolectic. They need to have Cucurbita to be able to establish in an area. So basically, we have been studying this system to kind of like understand if the current distribution of this squash bee has been driven by the domestication and cultivation of Cucurbita. So we've done some genetic studies to actually look for signatures of these population range expansion in the squash bees. And this was kind of like the main figure of like the first study that I published in this system. This was looking at the patterns of genetic diversity of squash bees across North America. These pie charts are indicating microsatellite markers that are genotyped, and the colors indicate the frequency of them. So you can see some patterns here, right? So in the southern and southwest, like part of the current distribution, we see more colors, right? Those populations are more genetically diverse. And then when we go to the extremes of the current distribution, we see a significant reduction in the genetic diversity of the bees, which is exactly what we kind of expected if the current distribution of the bee is the result of a recent range expansion. So, well, at the time, those markers didn't really allow us to date like these events. We didn't really know when this happened. So our working hypothesis was that the range expansion of the bee, particular to the East Coast, was facilitated by Cucurbita domestication. And so you may know or not a lot about Cucurbita domestication. Cucurbita is one of the first domesticated crops in the world. It was first domesticated here in Mexico about 10,000 years ago. But there was a second domestication event here around the Ozarks area that happened about 5,000 years ago. So, you know, our working hypothesis was like, okay, this origin of these, you know, like expansion to the northeast probably was facilitated by that domestication event. Well, so more recently, we expanded our genetic approaches, and we started, we sequenced the entire genome of the bee, and we genotype markers across the genome of the bee, and we use basically the same sampling from the previous studies, and we augmented it, so we included additional populations, and that ended up with over 100 haploid genomes that we genotyped with double digestion rat-seq, and then we did some full genome sequencing. And for this study, we wanted to have a better understanding of, you know, like the timing of these events to see how well we could link expansion of agriculture with changes in the demographic history of the bee. So the first hypothesis that we tested was that Cucurbita domestication not only increased or facilitated the expansion, geographic expansion of the bee, but it also facilitated increases in population sizes. So we can think that, you know, if there are more flowers, right, that's going to support more squash bees, and we would kind of like detect those changes, demographic changes in the current populations. And then the second hypothesis, which is something that I'm very interested in, is whether this transition from areas here in the Southwest and the Midwest, right, like to the Northeast, where they are exclusively inhabiting agricultural settings, is actually linked to adaptive processes, which is kind of like the first question that I posed. Okay, so I'm going to start showing you what we found in terms of just like patterns of genetic diversity, which I think is really interesting. And it may drive some people crazy in a few years. Because, well, this is the genetic composition of a squash bee, these species of a squash bee in North America. We find 5 clearly differentiated lineages, right? So we have a lineage in Mexico, then we have a lineage in the eastern part of the distribution. Most likely the center of origin of the species is here in what is currently Arizona, and then we have these, you know, like 3 lineages that very recently diversified, one in the Colorado Plateau, one in the Great Basin, and one in the Pacific Coast. So I have visiting a student from Belgium who is very interested in taxonomy and species delimitation, and he's working on actually trying to characterize whether what we see at the genetic level can be related to morphological differences. I just took some photos of the bees that are in our collection. Color is a terrible taxonomic character, right? Like, I'm not saying that those are different species because they have different colors. But like other characters like the genitalia, for example, are showing, you know, quite distinct differences. So we may have more than one species described in the next couple of years. And I know a lot of people are not going to like that, but it is supported by, you know, genetics and likely morphology. Okay, so let's go back to demographic history. So what happened with the population sizes of squash bees in North America? Well, another thing that I forgot to say is that, you know, like that diversification, the early lineages, right, which are Mexico, Arizona, and the eastern lineage originated way before humans were cultivating or domesticating any crops, right? So there were some early diversification, there was an early diversification of the bee. But then we wanted to know, because our hypothesis was that there was an impact of the cultivation of these cucurbita plants on the demographic history of the bee, we wanted to actually look at changes in effective population sizes in the past 10,000 years. Okay, so I have the lines there are indicating 10,000 years, the first domestication event of Cucurbita pepo in Mexico. And then this second event, which is, yeah, I thought it was 5,000, but it's probably like 6,000 years ago, which was in this area of Missouri. So now we're going to see some lines that are indicating effective population sizes over time for those lineages. And so basically what we can see is that, yeah, those three lineages are old, and then about 8,000 years ago, right, when in the beginning of the Holocene, we had this Colorado lineage originating, right? And then what we see is that this lineage in the far east of North America really originated much, much more recently, right? Like, so we're talking about events that are about 2,000 and 3,000 years old. So our initial hypothesis was not supported, right? Like the origination of this far eastern lineage is really linked to more recent events that we think are associated with sizable agriculture of Cucurbita in North America, and not necessarily the domestication per se. The other interesting thing that we see is that in the past, this is really hard to, there are error rates associated with all of these dates. But in the very recent past, right, like about maybe 1,000 years, during the past 1,000 years, we see this synchronized increase in effective population sizes in all of the lineages, right? So this hypothesis that the number of flowers at a continental scale had an effect on population sizes is supported by the data. Okay, now I have a little video to kind of like show you the changes in the distribution of the ancestral plant and the squash bees. So here we have in red the squash bee. This is the buffalo gourd, which is the common, the wild Cucurbita that is common in Western North America. And then we're gonna see, this is the time, right? So we start during the last glacial maxima. Remember that the entire continent was covered with ice, right? So the distributions of both the bee and the plant were pushed to the south. And then with the beginning of the Holocene, right, we see an increase in both the distribution of the plant and the bee, and this is likely when some of the diversification, for example, of the Colorado Plateau was facilitated. And then you will see that in the past 2,000 or 3,000 years, which is based on the figure that I just showed, the population of the bee, like branch off, likely because there was enough agriculture in the northeast of North America to support the expansion of the distribution of the bee. So it's a really, really cool story. Okay, so the second question was related to adaptive processes, right? So we can think that the ancestral environments where these bees were located are quite different from where we have them here in the Northeast, right? Like, so these are the plants of Cucurbita feridissima. Those plants are, like in the desert. They have very patchy distributions. And then this is how we do agriculture, right? And likely the agriculture like 1,000 or 2,000 years ago looked different, but probably it was, like plants clustered together, kind of like having a very reliable floral source year after year, which is quite different from the desert locations. So we don't know, this is one reason, but we can think many different aspects that are different about this environment, like for example, temperature regimes, right? There are many different conditions that make the ancestral and the agricultural environments different. So what we did is we did full genome resequencing for populations that are located within the ancestral range of the plant, right? Like, so basically have had these interactions with wild plants since the origin of the species. This recently derived lineage that is very close to the ancestral range, and then one population from Pennsylvania that is in the kind of like extended, very recent range. And what we did was looking at protein coding genes and looking at signatures of positive selection in these genes. What we found is that about 20% of all of the genes in the species show signatures of positive selection. And so this is just like Venn diagrams, like counting the number of genes where we found those signatures. And so we can see that, like both the population in Mexico and Western North America, they have some areas under selection, but really the majority of the selection is found in the Eastern North America lineage. And when we looked at the genes that show signatures of selection, what we find is this very interesting dominant pattern of genes that are associated with like sensory biology in the bee that are under selection, right? So this is kind of like a very coarse way of looking at the potential functions of these genes. We don't really know what they're doing, but they seem to have, like these kind of like functional categories in this Go analysis. So this is where we are with the story. One of my graduate students, Avehi Singh, is actually, like looking into this. So she's diving deeper into characterizing families of olfactory genes in squash bees. And we're finding this very, very interesting expansion in some gene families, OR gene families, in the Eastern North America lineage. We don't know exactly what that means functionally yet, but this is where we are. So I think with all of these studies, we have kind of like developed this very interesting system where we have shown that agriculture, humans have had kind of a huge footprint evolutionarily on pollinators, right? The agriculture of Cucurbita facilitated this expansion in space, this super exponential expansion in size. And in one of these populations is also basically this transition to a new habitat is facilitating adaptive processes that we don't fully know exactly, what they are associated with, but we have a hint based on, like the potential functions of these genes. There is something interesting happening that is impacting how the bees are sensing their environment. So, well, these are the conclusions for this section. Oh my God, am I driving you crazy with the? Sorry, I just noticed that. Okay. Okay, so I guess I'm going to come back to the first question that I posed at the beginning, right? Like, can pollinators adapt to agriculture? So I would say, like, maybe like a small, like, yes, right? Why do I say that? Because we do find this very clear evidence that the Eastern North America lineage has, these massive signatures of adaptation across the genome. Whether that is related, like directly linked to agriculture, remains to be proven. But as a general association and a general pattern, we see this, right? So one of the things that we would like to do in the future is actually use the same approaches to look at signatures of adaptation in other populations of the squash bees in the western part of the distribution, where the bee is also inhabiting agricultural environments exclusively, right? And so I think if we show that, like there are similar underlying processes in every kind of like new population of the squash bee, I would have a better, stronger yes, bigger yes to that question. But then the next question is, what is driving these processes, right? Like these adaptive processes. So I'm going to come back to this framework. And again, this is, I feel like this project is like my 30-year career, project. So I'm just going to like share with you, what we have been working on and how we are tackling this question, which is, like the first thing is, like whether we see actually some changes in the floral traits of the crops and some changes in the behavior of the bees that could be, like driving adaptation to agricultural environments. So, well, this is a project that was completed a couple of years ago, but isn't published, and it was led by Kristen Broshu, who was a postdoc in the lab, and she's now an assistant professor at the University of the Bahamas. And basically what she did was asking questions about functional floral traits associated with crop domestication, and then she followed that up with some field experiments looking at foraging preferences of the bees, comparing wild and domesticated Cucurbita species. So, well, as I was saying at the beginning, Cucurbita is a very, very nice system to ask comparative questions about domestication, because we have different independent origins of domestication within the genus. So this is a phylogeny of some of the species. They are not all here, but these were the ones that we included in the study. And so we can see that in orange we have domesticated species, and then in green we have wild species. So it's not a perfectly balanced design where you have wild and then domesticated, branching off. We actually don't know the wild ancestors of some of these lineages, but with the phylogenetic information, we can take into account, like the origins, the independent origins of these different lineages. So what we did is we grew individuals from all of these plants in the greenhouse, and we focused on characterizing different floral traits from these plants. So we looked at morphological traits. We characterized 12 of them. We also collected volatiles, and we quantified different nutritional metrics for pollen and nectar. So for pollen, we did carbohydrates, proteins, and lipids, and then we calculated protein and lipid ratios. And for nectar, we quantified glucose and sucrose. There could have been more done, but that's what we decided to do. And then we selected a subset of those 13 species, and we grew 7 of them in the field. We did a complete randomized block design. We had 4 wild species, 3 domesticated species, and for 2 years we looked at pollinator visitation to these different species of Cucurbita. We observed everything, but these communities are really dominated by 3 species. Squash species 1, Bombus impatiens, which is a bumblebee, is a generalist, is another one. And we do see some honeybees, but the honeybee abundance is not as high. So the data had a lot of zeros in some cases, so I'm not showing the honeybee data today. Okay, so what did we find with the functional floral traits? I'm going to show you some plots of traits in multivariate space, so like PCAs and NMDSs, and you will see the symbols in orange are domesticated individuals, and the symbols in green are wild individuals. Okay, so for morphology, what did we find? Again, this is domesticated, this is wild. What we found is that the flowers of domesticated and wild Cucurbita species are basically occupying different morphological trait space, and this was driven by a reduction. So we see these 2 traits that are important: anther length and corolla height. What we see is a reduction in the anther length and corolla height for the domesticated plants. So those tends to be like larger and wider in the wild plants. For volatiles, the main pattern that we see is that there is a reduction in the abundance and complexity of floral volatiles for domesticated flowers. So you can see here all of these domesticated, flowers of domesticated individuals, and then you can see that the wild species have more abundant and more complex volatile blends. For pollen, this was actually quite conserved, right? Like we do see some more variation in domesticated flowers, but yeah, it was a non-significant pattern. So that was especially the pollen lipid ratio was surprisingly conserved across species. And for nectar, we again see a reduction in the concentration of the sugars for the domesticated flowers. So here I'm just summarizing, and I'm focusing on the changes on the domesticated flowers compared to the wild ones. In the interest of time, because I think I have too many slides, so I am not going to show you all the details of what we found with the preferences, but I can talk about that in these plots. But basically what we did is we summarized all of the visitation information into kind of like an average of the average visitation that we saw per species over the 2 years. And then we are doing this hand wavy thing, and we're correlating that with the traits that we measured in the greenhouse, right? Unfortunately, doing this trait characterization in the field is a little bit noisier and more complicated logistically. So what we did is make that comparison between greenhouse and field data. And the most interesting results were related to how anther length correlated with visitation of bumblebees, and how volatile abundance correlated with nectaring of squash bees. So here's what we see, and this is why you see, like you see those error bars, right? Like, so there are errors associated with the measurements in the greenhouse and the measurements in the field. But what we see is that flowers that have longer anthers, right, tend to have lower bumblebee visitation. So bumblebees really love the short anthers, and these are found in the domesticated flowers, right? So I don't remember exactly. I think these were like Cucurbita maxima, and I don't remember, like these species, but they love Cucurbita maxima, which has, like these kind of like shallower flowers. And then for the volatiles, we see this very interesting pattern where the squash bees prefer or are more abundant foraging in the wild flowers that have more abundant and complex volatiles. So this is specifically abundance, and you see that like there is kind of a significant correlation. And in general, we have like an outlier here, but in general, what we find is that the domestication process, like you saw that in the previous figure, reduces the abundance of floral volatiles. So again, we see kind of like this association. In general, there was not a preference for flowers being domesticated and wild and preferred by the squash bees. What we really saw was an association between the amount of the volatiles and the visitation of the bees. So, well, what do we see? I think I feel pretty comfortable saying that crop domestication has indeed changed functional floral traits, and in kind of like, in some cases, in some very consistent ways, which is quite interesting. And in terms of foraging preferences, we didn't really see any changes in what the squash bees are preferring. Remember that these all happen in Pennsylvania, right, which is in the extended range of the bees. So one of the things that we're wondering is, how would this be different if we do the same experiment in the ancestral range of the bee? I don't have data to answer that question. But what we see is actually that the generalists do prefer domesticated flowers, right? They were favored by certain compounds that are volatile compounds that are more abundant in domesticated flowers, and they prefer the morphology of the domesticated flowers. Okay, so the take-home messages are that agriculturalization of landscapes, including both crop domestication and generalist pollinators, we see associations with changes in morphological, volatile, and nutritional traits of these crop flowers. That the changes in the flowers associated with domestication tend to favor visitation by generalist pollinators. And so I guess one of the working, like questions that we have, or something that we're working on, is whether there is an association between these clear changes in the profiles of domesticated flowers and the very kind of like marked adaptive processes that the squash bees are undergoing in their sensory systems. I don't have answers to those questions. That's, those are things that we're currently working on. And so, like I feel like in the next, like 5-10 years, stay tuned, like what I think the goal is to really have a better understanding of how these different drivers of selection may be associated with adaptive processes in these squash bees. And for example, something that we're trying to do right now is using these hoop houses to be able to do more controlled experiments. Unfortunately, squash bees cannot be reared in the lab. I wish I could have them like honeybees in little cups and do all sorts of things with them, but they are just not very amenable. So this is working pretty well for us. It has limitations because it really depends on kind of like the natural cycles of the bees. These bees are only active for about 8 weeks of the year, so we are limited to our experiments to only a short window of time, which is, this is why I say like probably 10 years before we can have answers to some of these questions. But I hope I convince you that squash bees are kind of a unique and kind of like ideal system to investigate evolutionary and ecological responses of bees to the agriculturalization of landscapes. And with that, I would like to thank the funding agency. So we've had funding from NSF and USDA to do this work. And this is an old photo of my lab, but we're all wearing our squash bee, like lab t-shirts, so I really like it. And yeah, it's like, any questions, I'm happy to take them. Thank you. Yes. Yeah, most are not, but some are, right? And so I think what is interesting is, like to think about the limitations of that trait-based approach that I was talking about at the very beginning, because I think squash bees kind of like check all the boxes of the things that should go extinct with landscape, like land use intensification. They are specialists, they are soil nesters, they're univoltine. And despite all of this, like they have been able to presumably kind of like, I think there's a compelling case to think that they, they are adapting to these changes. So I guess to answer your question, I think it would be really interesting, and I hope, like, I don't know if we would have like the bandwidth to build kind of a parallel system, but there are systems, there are species and systems, for example, linked to blueberries. There are lots of specialists, like that visit blueberries, and like we are in the native range of, like blueberries. I know, Cesar's group does a lot of work with that. So I think it would be kind of like a very nice parallel system to, like ask some of these questions. And actually, like I didn't show that figure, but in that grab paper, they looked at whether agricultural intensification has positive or negative responses in different, in the kind of like the tree of bees that visits. They did that in apples, I think. And there are some specialist andrenids, for example, that show the similar kind of like positive response to agriculturalization as well. So I think that are potential candidate systems that one could think of to like ask similar questions and actually see if this is generalizable at all. Yeah. Yes. Sorry.
Are there studies like currently being carried out by this lab, or like will be? Because you said it was a short range, and I don't know what the time period for this
Studies for what? Sorry.
Is this lab like currently doing or planning to conduct anything else, or was it just for like this series of experiments you told us about?
So this lab, you mean my lab? Oh, yeah. Okay, sorry. So what we are working on, like I think what our mission right now is to be able to better understand what is driving adaptive processes in squash bees, right? But I think it would be wonderful to have similar study systems, like developed in other interactions between crop pollinators and cropping systems to actually see if there are any generalizable patterns, right? Like, do we actually see that flowers, Cesar is doing a lot of work with, characterizing volatile changes in crop flowers. There seems to be also kind of a reduction, right?
We have, actually, we just planted a common garden of wild and cultivated blueberries. So I'm very interested in that question.
Yes.
In our system.
So yeah, so I think it would be, like this question has been studied. I wouldn't say we have all the answers, but it has received a lot of attention from the herbivore perspective because we all want to kill the pests, right? And they seem to love ag. And so, but I feel like we really don't understand the unintended consequences of agricultural practices and pollinators in kind of adaptive processes. So
I think yours is the only study that I know on pollinators and domestication.
Yeah, I think so. I don't like saying that because, I never know who, but yeah, it's the only one that I know of. Yes.
What method was used for estimating effective population sizes from the genome data?
So that is based on coalescence simulations, right? Like looking at expected patterns of genetic diversity compared to like coalescence simulations over time. So that requires a lot of like modeling. And actually, you know, like Nate Pope, who was the postdoc leading that, actually like he developed a novel technique to use recombination maps to make these inferences, and now he's doing a postdoc only working on method development for that. If you're interested, I can put you in touch with Nate if you want to like learn more details about how.
That would be great at some
Yeah.
point. Yeah, thank you. You probably have to redo a lot of it for halfway deployed.
Yeah, but and you know, I think this is one of the caveats and limitations of, you know, like that paper. I think a lot of people email me, oh, I'm sorry, tell the presenter to repeat the questions. I am so sorry, Stephen. The question was, how were the effective population sizes estimated for the squash bee demographic study? And so my response was based on coalescent modeling and recombination maps. What was the, oh no, I was saying the haplodiploid stuff. So yeah, so he basically developed a method to deal with the ploidy issue, but the stuff is not very well documented. Like his GitHub is kind of like, he understands and no one else. So, so yeah, I think it is great that he's working on methods development, because I think what it is important is now develop things that can be used by the community widely. Like, you know, I feel like I'm an empirical population geneticist. Like, I cannot develop methods, but if you develop something that we can use, right, like we would all benefit from it. So, I think you had a question too. I'm sorry. Just so
I was just wondering, is there any way to ascertain, or do you know if the domesticated plants, or if the domesticated curcubans, if they were spread before the, I guess, Pictonifus nebulosa, they caught up? Maybe that would be like a selective pressure on the curcubans to have shorter anthers and favor generalists?
Yeah, that's a great question. So the question is whether the spread of the cucurbita happened before the squash bees kind of colonized these agricultural environments. So the answer is yes, right? So for that paper, we also looked at records of basically the way this is kind of inferred is whether you have seeds that show, you know, like you find a lot of seeds in kind of like the soil and, you know, like fossil records. So whether there are seeds that show evidence of domestication, and there are very early records in like Maine, right, that even predated the second domestication event of Cucurbita pepo in North America. So the plants were moved around, and again, most likely, you know, like they were being pollinated by things that were not, you know, squash bees. So yeah, I think we kind of like indirectly know that the plants moved faster than the bees.
That's fascinating.
Okay, and the other question?
My question was about the morphology comparison between the cultivated and native plant. I'm wondering if you did any sort of like color analysis, either in like visible spectrum or outside of it. I know they're both like orange flowers, but you can kind of get.
Sorry, I'm laughing because we started doing that and we dropped that. Okay, so
Okay.
the question was whether for the morphological characterization of the flowers of domesticated and wild species, we actually quantified any color metrics. So we didn't. So we took photos in both UV and, you know, like regular kind of like light, and so. I think, yes, the regular light, they all show, you know, like they're all kind of orange or yellow, you know, orange. The UV patterns were, you know, like quite different. And again, we didn't quantify it, so I cannot tell you, you know, there was, you know, like a clear distinction. But I remember that, you know, like there were flowers, and I don't remember if this was, you know, wild or domesticated, but some flowers had very strong, you know, like UV markings, and some were like, you know, like more like pale. So that is definitely something that, you know, should be done. Yes.
So like, I was just wondering whether like, other than a lot of the traits that we've talked about so far, I think they're very directly related to them. The bees are at the site with the plants, and like, sort of related to their that. And I wonder if, do you think that there are any other possible like sorts of traits that we might also look for, like signatures of effects of domestication? Like I was wondering, like maybe like dispersal or something like that. If, like, is there reason to think that something about the way that these plants were, you know, moved into this new range would have other sort of selective effects?
Okay, that's a great question. The question was whether something beyond what we characterize in the flowers could have changed, right, in the bees as they moved, like and colonized these new areas. So I think yes, and I actually, like, if I can speculate a little bit, like I wouldn't be, like, I think it is possible, for example, that some of the signatures of selection in the sensory systems of the bees may be driven by, like the fact that, for example, the flowers, like have lower volatile kind of like abundance, right? So even though the other plants are in the desert and, like the distributions are patchy, the flowers have very, very strong signals, volatile signals. And so it wouldn't be crazy to think that maybe the bees had to like work harder to identify the flowers in the landscape because of, this domestication. Again, this is super speculative, right? Like I have no evidence that this is happening. But something that we know that has been reported in the eastern lineage is this kind of like foraging period of their life. Like basically they emerge from the ground, and the females do not start building nests immediately. They have like a week where they, supposedly are looking for where to nest and find floral resources. I don't know, like maybe this is something that, they had also in the ancestral range. Maybe this is a new behavior. Yeah, I think that there's lots of questions to, investigate in that, sense. So I think I like where you're going with, your thinking of, yeah.
Thank you.
Margarita, with the profiles of the volatiles, do you see losses in some compounds, or the differences are more like quantitative, or do you see qualitative?
Yeah, both. So dimethoxybenzene, 1,4-dimethoxybenzene, is not found in the wild plants. It's only found in the domesticated plants. And within the and bumblebees love that compound. And within the domesticated plants, there are some species that have higher abundance of that compound, and some that like have very little of it. So Cucurbita pepa, for example, those flowers barely smell like anything, or they're super like plain flowers. Like yeah, they like I feel like the bees and well, so and so we're doing some electroentenograms to actually see how the bees respond to these different profiles, and so the bees here in the eastern lineage do respond to 1,4-dimethoxybenzene. Even though they are not found in the wild flowers. We're trying to do the same experiments with bees from Arizona, but it has been challenging. But yeah, that's one of the questions, right? Like maybe are they detecting different compounds, as they have adapted to agriculture?
One more wintry lab question. What are we supposed to call the bee right now? Right at this moment, what's the right thing to call it?
Xenoglossa prunosa.
Okay.
Yeah, sorry, I didn't want to bore you. Like I feel like only the bee, people care about those things. There was a paper about 3 months ago. We reclassified the generic classification. I think it's going to be Xenoglossa for the foreseeable future. Now we can publish fact sheets, and the name is not going to change, I think. But yeah, it's kind of a pain, but it's been, Yes.
Is there any kind of reason to believe that the changes in the sugar content between the domestic and the wild plants were either due to like the plant trying to influence vegetation rates, or is it due to like agriculture kind of selection of just profiles of the plant?
Yeah, so the question is related to whether the dilution of the nectar in the domesticated plants may be a strategy of the plants to get more visitors. Yeah, I don't know. You know, I know of a couple of other papers that report changes in nectar. Nectar is kind of like a very plastic floral trait. And like there is this one study that I love that paper that demonstrated that the plants near apiaries of, you know, like this one wildflower have more diluted nectar like near the apiaries, you know, like, and if you like draw a transect like out of the apiary, right? So the plants respond to pollinator pressure. I don't know exactly, you know, like, I guess it's because, yeah, I don't know exactly, you know, like what is kind of like the intention of the plant with diluting the nectar, but it seems to be a trait, that a floral trait that, you know, like changes very rapidly depending on the pressures of pollinators. Sorry, we're over time probably.
Are there questions online?
No, I think I was just asked to please repeat the questions, and I apologize because I didn't do it at the beginning, but.
And we have more times for the students
Yes.
who are signed up for the class.
Okay,
So,
thank
very
you.
much.